the general idea is that this enzyme, with its little active site right here that bonds the substrates, when a reaction is catalyzed in biology, the first step of it is usually for those things to bond to the active site, or maybe more accurately, to bind to the active site, because we are again not necessarily talking about a chemical bond forming between the substrates and the protein that's going to do the catalysis. What we're really usually looking at is something in the way of attraction, and those attractions are going to be a lot like the ones that made the protein form in the first place. So for instance, if I'm looking at something like chymotrypsin, which is a protease, it's a type of enzyme that cleaves up proteins into amino acids. Chymotrypsin is going to hydrolyze this peptide bond right here, this bond between the carbonyl and the nitrogen, the amide that was made when this protein was originally formed. And its active site is a really interesting thing because its active site makes a point of accommodating the nonpolar and polar parts of its substrate. So it turns out that chymotrypsin is specific to to something that has phenylalanine as an amino acid. It only clips at that spot. And the reason is it has a little pocket in here of hydrophobic amino acid side chains, so things like alanine or leucine or heck, more phenylalanine, other things that are non-polar. And it's shaped right for phenylalanine's side chain to fit in here, and it's all oily. And so as a result, this slides in no problem. And on the other hand, over here where the carbonyl is located, you've got two peptide hydrogen atoms that bind to essentially to the substrate. So you've got a hydrogen bonding attraction forming between each of these. You've got the positively charged H's and the negatively charged O. Lastly, there's a serine side chain with this OH group right here that forms an attraction with the carbon, because remember that carbon is bound to an O and an N, so it is pretty electron starved, so you can stabilize its presence in there by providing an OH, particularly the O portion of it, which is negative and will attract the positively charged side of the carbonyl. So all of this is going to lower the energy of your substrate when it binds, and so that encourages it to go into the binding pocket. Now, worth noting, there are ways of adjusting that active site. The active site isn't going to, it's like any other molecule. It's going to be vibrating at all times. One thing that you can do is play around with what are known as effectors. Now, those are molecules that bind somewhere else in the enzyme. So bear in mind, we've talked a bit about the substrate binding straight to the active site. You can also play around with the shape of the structure by bonding in other places. Remember, if I bring in something negatively charged that suddenly pulls a chunk of the protein, well, that's going to move the stuff that's right there, but then everything in the next level in from there is going to get pulled and pulled and pulled. And so the result can be that a small little binding event in a different place on the protein can change the entire shape of the protein. And this can work in both ways. It can make something more effective as a binder, and we call things like that positive effectors. Sometimes you have enzymes that are made in your cells, the proteins are expressed by the ribosomes, but they aren't in the active state yet. They require some group to come in here, slightly change the shape of everything, and make an active site that can actually bind the substrate. That can be a very useful way of making a protein so you have it ready when you need it. And maybe in some cases the effector is even some of the substrate. So it's only when you've got a lot of the substrate around that the binding event happens and you wind up with an active enzyme. Okay, so that will increase your enzyme's activity. We call those sorts of things positive effectors. You can also have negative effectors, and they do the exact opposite. That's something that might shut off an enzyme. And they, again, will do the same thing. They'll bond somewhere other than the active site, but when they do, a change occurs in the overall structure of the enzyme. The active site doesn't look the way it did, and it no longer binds the substrate. Because this lessens the activity, this would be a form of inhibition. And we call negative effectors non-competitive inhibitors. Now, the reason for that is because you can inhibit another way too. You can do a very direct method where instead of binding something that messes with the shape of the active site, you can bind something that actually sticks in the active site and blocks it. So for example, this is succinate dehydrogenase, a little cartoon of it. This is one that you're going to see in the Krebs cycle, the citric acid cycle. Succinate dehydrogenase normally bonds, as you might expect, succinate. But if I have something with a very similar structure and very similar charges on either side, because succinate, in order to bond something with carboxylic acids, more accurately carboxylates on both sides, succinate dehydrogenase has a bit of positive charge here and here. Well, if I bring in something like, say, malonate, that would come in here and bond into that spot just as easily. It's got the right shape, it's got the right charges, but it can't react. What I wind up with is just a simple blockage of the active site. In fact, one thing worth noting is that the reason succinate dehydrogenase can do something with succinate, the reason it can dehydrogenate it, is because you have two carbons side by side right here that could each lose an H. And when they do, I would form a double bond. That would make something known as fumarate, which we will see, again, as one of the steps in the citric acid cycle. Malonate doesn't have the two side-by-side carbons. It's just got one. There's nowhere to put a double bond here. And so you're not going to be able to form the proper product. And so malonate here acts as a competitive inhibitor. It blocks the site, it doesn't react, and it prevents succinate from bonding in there and doing what it's supposed to. Not oxidizable, no adjacent carbons with H's, and therefore nothing interesting happening besides knocking down the rate of the reaction that we want to have happen here. While we're talking about things that can bring about higher or lower activity for an enzyme, let's chat briefly about these non-peptide groups that sometimes need to bond. So we saw effectors are things that generally bond elsewhere in the protein. Competitive inhibitors and cofactors, prosthetic groups like these, are going to be things that generally bond in the active site because they're involved in the reaction. One of those things that we've already talked about when we looked at chemical reactions back in chapter 6 are the coenzymes that are responsible for bringing and taking hydrogens when we do redox chemistry. We're going to talk about them in a lot more detail next week because we're going to be looking at metabolism in next week's lectures. But for now, it's just enough to know these are little molecules that are needed to actually carry out the function. So NAD plus or FAD, neither of these are made of amino acids or chunks of protein. They're just small organic molecules. They can be transiently bound. NAD plus is a good example. It jumps onto the enzyme when it's needed and otherwise isn't there. FAD, on the other hand, is bound to the enzyme. In fact, FAD is the thing that takes away the H's when succinate dehydrogenase dehydrogenates succinate. And it actually, the entire enzyme with the cofactor gets carried to the mitochondrial membrane. So you sometimes need these things in order to make a reaction happen. Another great example of these are the hemes that are bound to hemoglobin. That's a large organic molecule with iron at the center of it, not protein-based, but absolutely necessary for the function of the enzyme.